Now coming to the, as we have discussed about PCT in previous video. Okay, one more thing about PCT I want to mention here, that the fluid living PCT was tell what is its nature. It is isotonic. Okay, why it is isotonic? Because we have seen that the maximum part of solutes is absorbed in PCT, and we have seen also that 70%, 65 to 70% water is also absorbed along with the solutes. Okay, the solute and water both are getting absorbed in the same proportion, so there will be no change in the concentration, so that part of the fluid is isotonic. Okay, next we are going to discuss about thin ascending limb. Okay, we are going to about thick ascending thick ascending limb. Okay, I'm making this diagram. This is the lumen of thick ascending limb, and this is the cell, okay, of thick ascending limb. Again, the most important part here is that the presence of different types of uh, transporters. Okay, and as in the PCT, on the basolateral side there will be the presence of sodium potassium ATPase that will throw out 3 sodium outwards and 2 potassium inwards. Okay, this is the sodium potassium ATPase, no problem here. The thick ascending limb, the most important transporter of the thick ascending limb is this one, which I am going to show you. This is the diagram. Sodium is coming in, potassium is coming in, along with 2 chloride ion. Okay, this is the sodium potassium 2 chloride ion, or it is sometimes written as NKCC2. Sodium potassium 2 chloride, Cc chloride and 2. Okay, NKCC2 channel, and this is the characteristic channel of the thick ascending limb. Okay, mind this. This is the characteristic channel of thick ascending limb. NKCC2 channels. Okay, NKCC2 channel. Don't get confused and be perfect in remembering this, that this NKCC2 channel is associated with thick ascending limb. Okay, here sodium potassium ATPase is working. One more important thing is here, that the thick ascending limb is very important in regard with the transporters, and here this is very important. Again, sodium H plus anti-transport, as we have seen PCT. Okay, this is the most important, which I am going to draw. This is the potassium leaky channel. Okay, this is the potassium leaky channel. This the same channel are present in PCT on basolateral side, but in the thick ascending, which is present on the apical side, potassium leaky channel. Okay, and due to the presence of potassium leaky channel, potassium comes in lumen, and here it creates plus 8 millivolt potential in luminal space, positive charge plus 8 millivolt, and due to this generation of positive charge, this is the another cell, and due to the generation of this positive charge, other cations which are present in luminal space comes outside, such as calcium plus, Mg plus 2, and blah blah blah. Okay, and this Mg plus 2 is very important because it is the Mg plus 2 is maximally reabsorbed in thick ascending limb, so mind it. Okay, thick ascending limb in Mg plus 2 about. Okay, next we are going to see about some diuretics, that is mainly your loop diuretics. Okay, spelling is not correct here. Loop diuretics, and some of the example is loop diuretics are your butamide and something furosemide. Okay, this is butamide and furosemide. These two loop diuretics generally target this NKCC2 channel. Okay, this is the These are the two loop diuretics, butamide and furosemide. This will target generally NKCC2 channel. Okay, NKCC2 get blocked by these two. Okay, then what we will see, if NKCC2 will be blocked, then the osmotically active particle will not enter the cells of thick ascending limb. So this osmotically active particle will present in the lumen. Okay, and as we know that where there will be the presence of osmotically active particle, there will be water, so water will be present more in urine. Okay, so this is known as diuretics. It will increase the amount of water in the urine. Okay, by this way butamide and furosemide will increase the water in the urine. Okay, okay, and if there will be mutation in your ROMK, this ROMK is nothing, this is the ROMK. Okay, leaky potassium channel is also known as ROMK. Potassium sodium chloride, yes, this was the thing which I have missed to tell you about sodium. I'll leave this ROMK, or simply our NKCC2 channel, NKCC2 transporter. If there will be mutation in both of them, then that is known as Barter's syndrome. It is known as Barter's syndrome. Okay, and there is the three characteristic feature of the Barter's syndrome. That is the hypokalemia. Another one is ECF volume get decrease, and due to this decrease in ECF value, we will know aldosterone will be secreted, so there will be hyperaldosteronemia. Okay, and the third is increased H plus secretion. Okay, that is metabolic alkalosis will be there.